Hi everyone. Okay, so in this video I'm going to be talking to you about the process of transcription and translation. Okay, so as you know, our DNA is a code, and what it codes for is proteins. DNA is made up of nucleotides, and each of those nucleotides has a backbone made up of phosphate and a pentose sugar, and then the variable region, which is the nitrogenous base. The order of these bases is what makes up the code. So, in this code, 3 bases codes for 1 amino acid, and we know that proteins are made of amino acids. Students often make the mistake of saying that DNA is made of amino acids, for instance, or saying that DNA makes amino acids. DNA doesn't do that. DNA codes for amino acids in something that we call the genetic code. So let's imagine this is our double helix, and we know that our polynucleotide is made up of genes. We also know that genes are made up of introns and exons. Exons being the expressed region of the gene and introns being the non-expressed regions of the gene, the bits that are spliced out of the DNA before it's produced into mRNA. So when we think about the term gene, we can either use the word gene or we can use the word cistron, and cistron is just an intron and an exon joined together. So all of our DNA is split up into genes. So let's imagine that we just wanted to read a particular gene because we wanted to produce a particular protein or a particular polypeptide chain. In general, remember, it's better to say that genes code for polypeptide chains as opposed to proteins, because some proteins are made of more than one polypeptide chain. So here we are inside our nucleus, and this is where transcription is going to happen. And transcription is the production of a single-stranded piece of mRNA taken from the one section of DNA. So the first thing that happens is the enzyme DNA helicase acts on the gene or cistron to break the hydrogen bonds between the bases, exposing the bases on the 2 strands of DNA. So DNA helicase unwinds and unzips the DNA, and it produces what is known as a transcription bubble. What that means is a section of DNA in which the bases have been exposed, and this section of DNA codes for 1 polypeptide chain. Okay, so now we have exposed our bases on the template strand. What happens next? Another enzyme comes along. This enzyme is called Rna Polymerase, and it binds to the template strand of the DNA at the beginning of the sequence that is going to be copied. At the same time, free RNA nucleotides that are present in the nucleus. And let's just have a think about what an RNA nucleotide is. Okay, so an RNA nucleotide is a nucleotide very similar to DNA. So it's got the phosphate, it's got the pentose sugar, but instead of having deoxyribose as its pentose sugar, it's got ribose. And the bases are different. So we know that DNA has adenine, cytosine, guanine, and thymine, but in RNA, instead of thymine, there's uracil. So we've got these differences. We've got many millions of these RNA nucleotides floating about within the nucleus. So I'm just going to make this diagram a little bit bigger so we can actually see what's going on internally at the template strand. I'm also just going to delete the non-template strand just because it's going to get in the way of our drawing. Okay, but it hasn't gone away. I just am going to rub it out so that we can see what's going on the template strand. So like we mentioned before, there are millions of these free-floating RNA nucleotides that are present within the cytoplasm, and so I've just drawn a few here. So these will come along and they will form hydrogen bonds with their complementary DNA nucleotides. So with our A U will come along and it will form hydrogen bonds. Then another U, and a A, and a C, and a G, and a G, and a G. Right, so these free-floating RNA nucleotides will come along and form weak hydrogen bonds. No enzyme is needed for this to occur. What the RNA polymerase is needed for is the formation of the phosphodiester bonds between the RNA nucleotides. So as the RNA polymerase moves down the strand, it catalyzes the formation of the phosphodiester bonds between the backbone, creating a continuous backbone between the RNA nucleotides. At the end of the cistron, or gene, the RNA polymerase will reach a stop code. This will cause the RNA polymerase to stop moving down and to break off, and at this point the production of pre-mRNA is complete. As all this is happening, so the DNA helicase was going down and unwinding and unzipping by breaking the hydrogen bonds, and the RNA polymerase was following closely behind, causing the formation of the phosphodiester backbone on the RNA strand. As this is happening, just behind that, the DNA is rewinding and rezipping and reforming its hydrogen bonds between the RNA polymerase. And now the pre-mRNA detaches, and we have a single strand of mRNA in the nucleus, and we have our double-stranded DNA. Okay, so I've just cleaned up this diagram a little bit. And I've obviously changed the code here just to make it a little bit longer. So I just want to talk to you a little bit about terminology. So 3 bases in DNA is known as a triplet, whereas in RNA, 3 bases is known as a codon. So we can see in what we've drawn here, our section of RNA is made up of 4 codons. Okay, so we've got 4 codons. Okay, so now what we have here is pre-mRNA. So we know that pre-mRNA needs to be spliced to have the introns removed, and it's spliced using enzymes to form mRNA. So that's the final stage of transcription. Once the mRNA has been spliced, it leaves the nucleus via the nuclear pores. So there are little gaps in the nucleus called nuclear pores, in the nuclear envelope called nuclear pores, and the mRNA leaves via these gaps. And that's it. That's the end of transcription. So now we're going to move on to the next stage. The next stage is translation, and translation occurs on the ribosomes in the cytoplasm. So a couple of key things to note about this tRNA molecule. So can we see here that it says it's made up of one folded polynucleotide chain, and it's the shortest of the polynucleotide chains, made up of 75 nucleotides. Now it's a single chain, but it's folded, so it does contain hydrogen bonds. Now, if we think about the important parts of the tRNA molecule, the first important part is this bit at the bottom here, which is called the anticodon. The anticodon is a set of 3 nucleotides that is complementary to a particular codon. And remember, a codon is a set of 3 bases on the mRNA. So it's this section that is going to bind the tRNA and the mRNA together via hydrogen bonds. And the second important part is the amino acid attachment site. So when you have amino acids in your bloodstream and you absorb them into your cells, those amino acids are floating about in your cytoplasm, and then they get picked up by a specific tRNA molecule at the amino acid attachment site. And there is a different specific tRNA for each amino acid. What that means is the the anticodon, so whatever this anticodon is at the bottom, will determine which amino acid will be bound at the top, so that the codon on the mRNA will ensure that a particular amino acid is going to be added to the polypeptide chain. So, the mRNA has left the nucleus via the nuclear pore, and once it's left the nucleus, it travels to and binds with the ribosome. At the ribosome, there's an mRNA attachment site, and there are also tRNA attachment sites in the small subunit, and that's going to become important later. So you might have noticed that I have changed the mRNA strand. Don't stress about it. We'll stick with this one from now on. So the first three exposed bases are always AUG, and AUG always codes for methionine. In order to understand that, we need to have a look at the genetic code table. So let's take a look here. The code is really simple. So if the first base in the mRNA triplet is A, and the second base is A, and the third base is A, we get lysine. So A gives you the amino acid lysine. But then look, so does AAG. That also gives you lysine. So if we had the first base was G, the second base was G, and the third base was G, we'd get alanine. But also if the first base was G, second base was G, and third base was A, or C, or U, or G, still alanine. So you can work out which amino acid it is just using this table. So if we had C, 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 we'd have proline. You can also see that some of the codons don't code for amino acids. Can you see here you've got a stop codon and another stop codon here? So if you had UAA or UAG, or you had UGA, this wouldn't code for an amino acid. It would just denote the end of the mRNA chain. So we'll come back to this table in a minute, but this table is really important in understanding how to read our mRNA. So let's just take a minute to compare it with the strand that we have and see if we can work out what amino acids our mRNA chain is coding for. So let's take a look at the first one. So the first one is AUG, and we can see AUG codes for methionine. And the next one is AAU, and we can see that that one codes for asparagine. And the next one is AAG, which codes for lysine. And the next one is UAC, and that codes for tyrosine. And the next one is CAU, and that codes for histidine. So this is important that we can understand how the genetic code is used to determine what amino acid is coded for by each codon on the mRNA and triplet on the DNA. Let's talk through the process of translation. So the mRNA has moved from the nuclear pore to the ribosome, and then a ribosome becomes attached to the starting codon at one end of the mRNA molecule. A tRNA molecule with a complementary anticodon to the first codon on the mRNA attaches itself to the ribosome. So in this case, the anticodon is going to be UAC, and the amino acid binding site on the tRNA is going to be attached to the amino acid methionine. When this happens, another complementary tRNA molecule with the complementary anticodon to the second codon on the mRNA attaches itself to the ribosome at the next attachment site on the ribosome. So let's just have a look at that. So when this occurs, this is bringing these two amino acids in close contact with each other, and they form a peptide bond. Once this peptide bond has been formed, the first tRNA molecule leaves the ribosome, and when it leaves the ribosome, it leaves its amino acid behind because it's attached via the peptide bond. This tRNA molecule will then just go off and pick up another methionine free floating in the cytoplasm. When this occurs, the whole mRNA chain moves through the ribosome, and another tRNA molecule comes along and binds onto the next codon. And another tRNA molecule comes along and binds on at the tRNA binding site within the ribosome with an anticodon that is complementary to the codon on the mRNA. So this time it's going to be AUG. And this process is the ribosome. And coming off the ribosome, we are forming a big long chain of amino acids all joined together by peptide bonds. Now, the mRNA strand is moving through the ribosome. This mRNA strand moves through one ribosome and then enters the next ribosome. And as it enters the next ribosome, that ribosome starts to read it. And as it starts to read it, it produces another strand of amino acids, the same polypeptide chain again. And this happens over and over again. So the mRNA strand moves through the next ribosome, and that ribosome starts to read it, and it moves through the next one, and that one starts to read it, and so on, and so on, and so on. This process of the mRNA chain moving through many of the ribosomes and being read each time to produce a polypeptide is called a polyribosomal system. So one mRNA strand can be read many times by the ribosomes to produce many polypeptide chains, each one identical. And this is important because, say for instance, you needed to make some amylase. You don't want to make one molecule of amylase. You want to make hundreds of thousands of molecules of amylase, and you want to make them fast. And you don't want to have to make 100,000 strands of mRNA in order to make 100,000 amylase molecules. That's incredibly inefficient. So you just read the same set of instructions over and over again. So once you've made this polypeptide chain, this is the primary structure of a protein. So they're only joined together by peptide bonds. This polypeptide chain will then move through the rough endoplasmic reticulum. And one thing to note is I've sort of rather simplistically drawn this as a single line, but we know that it is a flattened envelope, right? So I know that looks horrible now, just live with it. So the polypeptide chain moves through the RER and then it pops out in a vesicle at the end. As it moves through the rough endoplasmic reticulum, it undergoes folding. So it goes into its secondary structure where it gets hydrogen bonds and its tertiary structure where it gets disulfide bridges. And then it travels in the vesicle. And where does it travel off to? It travels off to the Golgi body. And when it arrives at the Golgi body, here the vesicle fuses with the Golgi body. And at the Golgi body, it will undergo further processing and modification. So, for instance, it might be converted from a tertiary polypeptide chain, from a tertiary protein into a quaternary protein, or it might have prosthetic groups added, like it might be converted into a glycoprotein or a lipoprotein, or all that sort of thing. And so then it moves through the Golgi body, and then it pops out as a vesicle. And then that can either be a protein that's used intracellularly, or it might be sent towards the cell membrane where it would fuse with the cell membrane and be exocytosed out of the cell. So that's the process of translation. Binding with the ribosome, the tRNA bringing the amino acid the correct amino acid depending on the complementary pairing of the codon and the anticodon, the mRNA moving through the ribosome and the tRNAs detaching and leaving after the peptide bond has been formed, and then eventually reaching a stop codon, and then the whole polypeptide chain separating off, moving through the RER, and then heading off to the Golgi for packaging and modification. So that was a lot of stuff. That was a lot of process. What do I want you to do now? I want you to go through the process, make sure you can determine all the stages in transcription, make sure you can name the function of each enzyme and the name of each enzyme, and then translation, what occurs in what order, and make sure you make a list of all the new key terms that you've learnt in this process. But well done for listening to that, and I will speak to you soon.